Hello, my name is Marom Bikson, and today I'll be talking about how tDCS polarizes the highly folded cortex. Now, this presentation is for people who are really interested in the mechanisms of tDCS, the physics of tDCS, and sort of getting under the hood about how tDCS works. If you are a newcomer to transcranial direct current stimulation, tDCS, I and others have other videos that would probably serve as better introductions. This video assumes you know a bit about tDCS already, but does not assume you know anything about the engineering or the physics of it. I'll be trying to explain things as simply, but also as precisely as I can. All right, so this story actually begins a long time ago in 1968 with very pivotal work by Rush and Driscoll. And one of the things they did in this very compelling report is they modeled how current enters the brain. Now, at this point in time, they had to rely on very simple analytical models. And in this case, it was just a three-sphere model. So the head was represented as a sphere, with the outer sphere being the scalp, then the skull, and then the brain. Nonetheless, this work was very influential in helping us understand how current applied on the outside of the head reaches into the brain, and specifically, where does the current flow and how much of it actually reaches the brain. And this question remained an interest to people working in computational modeling, especially as the modern technique of transcranial direct current stimulation started to emerge in tDCS. Direct current is placed outside of the head, and it was very interesting to practitioners of tDCS to understand where does the current go and how much of it reaches the brain. And so you had work by Miranda, which used finite element methods, numerical techniques, using concentric sphere models, work from my lab as well, led by Abhishek Datta, where we started to ask questions about different types of montages, and I'll be discussing that as well, all the way to the development of essentially a closed form solution for these types of analytical models. So let me talk a little bit about this work that we published in 2008. Again, you can see that we are modeling concentric spheres. In this case, we have more layers, including the cerebral spinal fluid between the brain and the head. And you can see that we're modeling different montages. I know there's a lot going on in this figure here, but we're looking at two electrodes that are far, two electrodes that are close, an electrode surrounded by a very big return belt, and the introduction of a ring electrode, where you have one small electrode and then a ring around it. And each one of these pictures, what you're seeing is a simulation of how current from the outside of the head penetrates into the brain. And again, this is using concentric spheres. So let's focus on this one example, which is a bipolar electrode stimulation on the surface of the head. This shows you where the current is flowing. Now, one of the things we started to understand, even through these very basic models, is that when current enters the head, and it enters at the electrode we call the cathode, and then it exits the head at the current electrode called the cathode, it must complete a complete path. What that means is the current cannot enter under the anode, disappear essentially, and then reappear under the cathode to exit. Now, this may seem obvious, but it was an important conceptual thought in the context of transcranial direct current stimulation, where often people would put electrodes on the head and they would just focus on what was happening under the electrodes, but current must be conserved. And so current must flow from one electrode to the other. And so when you're thinking about how tDCS produces current flow across the head, you can't just think under this electrode and under this electrode. You must consider the path between them. And so that's what's being shown in the top image, where the entire region between the electrodes, not just under them, is being activated. The other thing we started to think about at this point was about the direction of current flow. So you can see that under the anode, which is the positive electrode, this is where current enters the body, and therefore it enters the brain. Under the cathode, which is the location where the current exits the body, it's therefore exiting the brain as well. And so what we are showing is that under the anode, you have inward current flow from the perspective of the superficial brain, the cortex, and under the cathode, we have current exiting relative to the superficial part of the brain. Now the directions that we talk about here, I'm going to be calling the radial directions. All right, so here we have radial, and here we have radial current as well. Between them, where it's flowing sideways relative to the brain, I'm going to be calling that tangential. And so now you can see that we can plot, first of all, where all the current is without thinking about direction. Here, we're plotting where is it flowing in and out. And here you see it's flowing in and out under the electrodes, not between them. Where is it flowing tangentially between them? We can see that that's between the electrodes. So this cartoon here is actually a pretty reasonable representation of what's going on here. But I can also plot something called radial directionality. So in this plot, a very hot color is radial in, and a dark color is radial out. So you can see we have radial in, and then we have radial out. And this is just a cross section. So looking at this, all these plots I should have said are the surface of the sphere, and this is a cross section as well. So this cartoon here is giving us an intuition about what the computational models are telling us. Now, why does directionality matter to begin with? Well, directionality matters because directionality is built into the structure of your brain. It's built into the structure of the cortex. It's been known for a long time. These are old studies from Cahal of cortical neurons, that the neurons in your cortex are not just oriented randomly, but they have a very structured column-like organization where they have these somas and these long dendrites pointed towards the surface. So these are the actual stains, and this is the cartoon I'll be using to illustrate this idea. Here's the soma, little dendrite here, big dendrite called the apical dendrite pointed upward. And so the cortex itself has a very directional morphology, and this ends up being very important to understand why tDCS affects the brain and how tDCS affects the brain. And so when we think about what tDCS does to the brain, we need to realize that the cortex has a direction, and so do the current flows themselves. So now what I'm showing you is the anodal under the red current direction. Remember, this is heading from the surface of the brain into the brain. Under the cathode, radial direction, but heading in this case out of the brain, and this would be the tangential direction shown here. And it turns out that each of these have a different influence on the neurons as a direct consequence of how they are polarized. A lot of these concepts, by the way, I'm going through maybe a bit quickly in a very high level. I've certainly dug into them more in my other videos, and I'm providing references for people who really want to get into the specifics. This is roughly how we imagine the current flow direction influences polarization. So when we have no current flow, we have no polarization by the current flow itself. In the anodal direction, which is heading down from the surface, we produce a somatic depolarization and an apical dendritic hyperpolarization. In the cathodal direction, which is opposite, we produce a somatic hyperpolarization. And when the current is flowing sideways or in the tangential direction, we do not produce a relatively significant amount of polarization in this axis. And this is something that has been modeled for a long time and was also demonstrated experimentally. So right away we see, right, that the direction of current flow has a big impact on how you polarize these specifically oriented cells, and therefore how you influence them. But it turns out that there are other structures in your cortex that are oriented differently. For example, there are synaptic inputs coming from other cortical regions and from deeper brain regions. And when these arrive in the cortex, they can arrive in all kinds of directions, including in the tangential directions. So we have a situation where we have these primary, called pyramidal neurons, pointed up and down, but we can have some axons coming in that are pointing sideways. And because of the way they are oriented in this case, they will not be polarized in these cases, but tangential current will polarize these axons and specifically these axon terminals. So the tangential current is not inert when we think about it in terms of the inputs that are coming in. And so this starts to explain why we don't just think about peak, but we're also looking at radial, right, in and out, and also radial direction. So where the inward current flow is, which is under the anode, where the outward radial current flow is, which is under the cathode. And we can also think about where the current is flowing sideways, and this is the tangential as well. And really what we did in this paper is we looked at all these different montages and we asked questions about how do they affect not just the focality, but the directionality. So you can see here that the ring montage, which is the one where you have one center electrode surrounded by a ring electrode, produces rather focal current flow, and is also more unidirectional. So you can see here we just have this black dot, a more unidirectional current flow, and I'll get back to that and why we find this montage particularly compelling. Okay, now, in 2007, 2008 we developed, and in 2009 we published a paper where for the first time we had an anatomically realistic head. And the way we did that wasn't by working with concentric spheres and imagining that the head is a bunch of spheres, but actually starting with MRIs, images, anatomical images of a human head, and then segmenting the scalp and the skull and the CSF and the brain into its various compartments. And they don't exactly look like a sphere, right? They look rather different. We also stimulated this head using realistic electrode shapes. So in this case, you see two large pads, and this is what was then the very common M1 motor SO supraorbital montage for tDCS. You can see it there. Now the electrodes are on the scalp, but I'm making the head transparent so that you can see the brain under it. So we stimulated that montage, and we also simulated something new that we had envisioned at that time, which was called the 4x1 high definition montage. And what did we find? We found that when you simulate the M1 SO montage, well, first of all, you will notice right away that the current kind of seems to be everywhere, right? It's certainly not right under the electrodes, right? It's under them, but it's also all the way between them. And you'll notice that the hot spots, which are shown here in the hotter colors, are also not under the electrodes, meaning the peak current flow delivered to the brain by the M1 SO montage is not necessarily even under the electrode, but you have all these clustering kind of hot spots. And you'll notice that these clustering little hot spots, right, these sort of yellow regions, seem to relate a bit to the folding of the head. So somehow the folding of the head is impacting where you get these hot spots. In contrast, the 4x1 montage produces a focal current flow. It has one center electrode and four outers, and the region of spread is actually controlled by the position of the four outer electrodes. At the time, actually, this was considered a rather controversial finding. Now I think it's pretty well established that when you use two large electrodes, the current doesn't just flow under them and doesn't exist in between. It must form a continuous path between them. And as that continuous path travels through this wobbly, gyrated head, it creates these clusters of hot spots. And as it turns out, the peak electric field or the peak current density in the brain is not under the electrodes, but it's going to be somewhere in between at one of these hot spots. We can take this a step further and represent not simply where the current is flowing, but where is it flowing in and when it is flowing out. Radial in and radial out. So in this case, radial in is shown in red, radial out is shown in blue, and 0 is represented in green. All right. This is a slightly different montage where we have an electrode at the top and a return on the side. But I'm using this to show you something, that the polarization you see is not like maybe what you might expect, which is a lot of red here and a lot of blue here, right? A lot of red here and a lot of blue here. Rather, what you're seeing is almost like a zebra pattern, right? Where there's some red and there's some blue and there's some red. And what's actually happening is that the current is traveling through the brain and it's cutting in and out and in and out of these folded gyri. What you see, in fact, because the pattern that it flows actually can change a bit as you rotate the head and look at it from different directions, or from the top, we're seeing a little bit more of the in, but from the side, we're seeing a little bit more of the out. But the point is that because of the folded nature of the brain, we do not see what we saw with the spherical models. With the spherical models, what we saw was under the positive anode, inward, under the cathode negative, outward. But because the head is so folded, that breaks down. Under the anode, we have a mix of in and out. And the same thing goes for under the cathode. And you could visualize it another way. Here now, I'm stimulating with just 2 small electrodes. Here's the positive, here's the negative, and I'm looking at what's happening in a single gyri, a single nodule between them. And you can see what happens as the current travels. It enters the brain, it hits the gyri one way, and then it exits the gyri the other way. What that means is that on this side of the gyri, so by the way, this is between the electrodes, right? It's not near the anode, it's not near the cathode, it's in the middle. What does this gyri see? On one side of it, it sees radial inward. And on this side, it sees radial outward. And this is an idea I really want to try to get across here, so I'll represent it in other ways. This is a different representation. It's another computer simulation where we're showing you where the current is flowing, and we're presenting it in terms of arrows. So look where the arrows are pointing. On this side here, so the surface in this case is this way, and we have radial in going this way. Here, the surface is on this side. We have radial going that way. So this is radial in, and this is radial out, and we have some tangential here on top. And so you can see why the fact that the brain is so folded inevitably creates this sort of alternating in-out pattern as the current washes through, okay? The same idea shown here. Current coming in, current coming out. So we do not have a situation in the highly folded human brain where we just got in and we just got out. It's always a mixed pattern. And so the question that arises from this is, all right, well, earlier I suggested that what happens to the brain depends on the polarity. I said, well, current flowing in has maybe one kind of effect, current flowing out has another kind of effect. Well, now we have both, right? You have some in and some out, and some in and some out. And the question is, do they cancel each other out? Is it sort of a zero-sum game? Or is there still going to be an effect on brain function? And the answer is there still is going to be an effect on brain function. And that's really, now that I've set up the problem, the solution to that problem is really the second main point of this presentation. So we need to go back to this idea, right? That when we're applying stimulation, we have this organization of the cortex, right? And we can have current flowing in, current flowing out sideways, and in some cases, both in and out, not in the same part of the brain, right, to be clear, but in adjacent parts of the brain where it inverts. How do we understand how this works? How do we understand how this works? So we want to imagine that we have a little piece of the cortex right here with our pyramidal neurons oriented in a particular way. We're applying direct current stimulation to that little piece of the brain. Now, that piece of the brain can be thought of as a little processing unit, and at the core of that processing are these pyramidal neurons. They receive inputs from other parts of the brain. could be other parts of the cortex, could be deep brain regions. And they integrate that input, and based on that, they provide an output. So this is your sort of neuroscience 101, how the cortex is functioning. And so when we apply direct current stimulation, we need to think of it as direct current stimulation of these cortical columns, direct current stimulation of pyramidal neurons processing information. And so when this direct current stimulation produces a polarization, in this case the anodal direction, producing a soma depolarization and a apical dendrite hyperpolarization, the question really is, what does that do to the information processing of this piece of cortex, right? So normally that piece of cortex is receiving inputs. It is deciding what to do with those inputs, right? Maybe it's these inputs generate an action potential, maybe they don't. And so again, the operational question for us is, when there is direct current stimulation, do we change how that cortical column is processing input or not, right? So that's this question right here. How does direct current, tDCS in this case, change cortical processing? So I think that is a very good way to frame this question in general to understand how tDCS works, but also to address this question of mixed polarization. Okay, so this is one example of how we assess this issue. Now, I know there's a lot on this slide, but let me break it down to its core components. We have an animal model, in this case a brain slice. And this brain slice, like a cortical column, in this case that's hippocampus, but in the same principle, is processing input. Input is arriving at these cells, they are processing that input, and then they are generating an output. In this animal model, we can measure the strength of the input, the strength of the signal coming in, and we can measure the strength of the signal coming out. Okay. Generally, what does that relationship between the input and the output look like? It looks like this. So here we have the input, and it's called the synaptic input, and it's coming in. And as the input starts to increase at a certain level, we start to produce an output. And as we provide more and more input, we get more and more output, and at some point it's saturated, we reach a maximum. And this is called an input-output relationship, or a neuronal input-output relationship, neuronal I/O, because here we have the input, and here we have the output. The relationship between the input and the output is what these cells are doing, right? They're converting the input to the output. Okay, so that's the first point. Now, in this experiment, we wanted to distinguish between two alternatives of what direct current stimulation can do. I'll explain each one in turn. The first hypothesis was that the direct current stimulation makes the inputs somehow stronger. Meaning normally the inputs have a strength of 1, now those inputs have a strength of 2. What would that do? Well, normally we would have this size input, but we've made the input stronger, right? So now this is the new input. Now this input is associated with a bigger output. Perfect. What if this was the initial input, right? Before we applied stimulation. Okay, we make that input stronger. Well, now we have a stronger input, so that's going to lead to a corresponding bigger output. Now this line here and this line here are the same line because the relationship between the input and the output has not changed. The processing essentially of these neurons, how they're working, doesn't change. They're just seeing a bigger input because somehow the direct current stimulation creates a bigger input. And there is a basis, by the way, to think about that. Here's the alternative. Not only do we make the input stronger, but we change the input-output relationship. So in the black, you see the original input-output relationship, how the cortex is processing information. And in the green line, you see the new input-output relationship, the new way that the cortex is processing information because of the direct current. Now what happens in this case? We apply the direct current, we will make the input stronger, right? Both things happen, but now this input is associated with this output. If this was the initial input and the initial output, so the initial input was here, the initial output was here, we're making the input stronger and we're making the input output stronger. What I'm trying to express is that in this case, the input-output relationship doesn't change. I mean, we move, it's easier for us to go down in this direction, but we're still living on this line. But in this case, the input-output relationship has changed. And we can measure that in this model because we can measure both the strength of the input and the strength of the output using various electrophysiological measures that I don't want to unpack here to keep it at a high level. You can certainly find the paper here. So what did we find? We found that when we applied the anodal variant of stimulation, and that's what's shown here. So this is current flowing into the brain. We changed the input-output relationship, shown here as I/O. Specifically, we made the whole system more sensitive. So that's what this line means. This line means that before, this input might produce a very small output. Now this input produces a very massive output. The direct current is changing how the cortex is processing information, effectively making the cortex more sensitive to a given bit of information. That's what we found on the anodal side. For the tangential current flow, where the current is flowing sideways, we found no effect on the input-output. Again, I'm simplifying it, but to first approximation, in this case, we did not report a change in input-output. When current was flowing in the opposite direction, okay, we found that there was a muting of the input-output response. Inward, soma depolarizing, enhanced input-output. Outward, soma depolarizing, soma hyperpolarization, muted input-output. You can say, okay, zero sum? Not exactly. Because the effect that we saw here with the anodal variant was much stronger than the effect we saw here. And this was explained in this paper. It's a function of the nonlinear properties of the cortex. Why is that important? Why is it plotted in this way? If I average all these plots here, right? So I say, what is the average change? I get this relationship here. I get more boost here than I get suppression here. And so even if things are entirely even, and I have 50% area of inward and 50% area of outward, the net effect is a overall increase in the input-output of this brain region. And that was demonstrated. So it is not a zero-sum game. Even if I have a piece of brain and it's half inward, half outward, I will see a net change in excitability. Now, obviously, it may not be that way, right? I may have a piece of brain where it's 80% inward and 20% outward, and then I'm going to see even a greater net increase. Or I might have a piece of brain where it's 90% outward and 10% inward, and therefore I may not see any increase at all. So where I land here obviously depends on the specific gyro I'm talking about, but it's not a zero-sum game. So that's one very important idea. Another important idea that I'm only going to mention, try to describe it at a very high level, and I have another video on oscillations and specifically tDCS and oscillations, and I have also explained how tDCS affects oscillations. But very briefly, here again we have a situation where we're stimulating the head. Here I'm representing the direction of current flow. And you can see again this notion that you have in and then you have out, right? And so in any given part of the brain, I have inward and outward. I don't have, in a folded brain, I don't have any region where it's all inward or where it's all outward. So that's what's represented here, okay? It means also that if I were to look at the neurons here and the neurons here, I have a population, right, that might be depolarized. And I have a soma depolarized, and I have another population next door that is soma hyperpolarized. These were deliberately not made the same size. Why is that? Well, maybe in this particular gyri, 30% of the gyri has current flowing in, and 70% of the gyri has current flowing out. And so because of that, it's not an even split. There's a disproportionate amount being polarized one way or the other. What we showed in this paper, which in this case was specifically interested in how stimulation might affect sleep oscillations, but the concept is very general, is that these neurons are not isolated, but they're tightly coupled to one another. This is where oscillations come from, meaning they work as a population. This one influences this one, and this one influences this one. So now imagine a situation where 70%, right, are in this hyperpolarized region, and 30% are in the depolarized region, but they're connected to one another. What this paper showed is that you can produce a net effect. Essentially, the majority wins, all right? Because they're all coupled to one another, if I excite, for example, this group and inhibit this group, the net effect on all of them is an excitation. Because they're coupled to one another, if I net inhibit this group and I net excite this group, the net effect is inhibition. I should note here the terms here, inhibitory and excitatory, refer to the type of interneuron, not whether they're being polarized one way or the other. So this is another, I want you to just think about this here at the conceptual level. If these neurons are all connected together and they function as a group together, if I polarize a majority of them in one direction, I can produce a net effect at the population level. Okay, one more idea of where we might see this sort of not zero-sum game, and this is when we're talking about plasticity. Plasticity is a process that all our brains undergo all the time, where connections between two populations of neurons can become stronger, all right, or can become weaker. When they become stronger, you might call it potentiation. When they become weaker, you might call it depression. And in an animal model, again, this isn't a data from animal models, you can produce plasticity, which is an analog of learning, a cellular analog of learning, by using something called theta burst stimulation. And I don't want to get too much into details here, but I want you to understand that when you have these two populations in an animal model, we can apply theta burst to them and produce a potentiation of the connection between them, a long-term plasticity, a positive plasticity, and that's what you see here. Hopefully you can make out the black points in the back. Okay. If at the same time as they are undergoing plasticity because of this theta burst, we apply direct current stimulation, tDCS, we can alter how much plasticity you get. Specifically, the anodal variant of tDCS, which is shown here as positive, gives you more plasticity. It is not that the direct current stimulation initiated the plasticity. The plasticity was ongoing because of this theta burst stimulation. It's that the direct current stimulation gives you, again, more bang for the buck, more plasticity for the same input. Cathodal variant, shown here negative, suppressed how much plasticity you get. So once again, the cathodal didn't generate the plasticity, the theta burst did, but the cathodal suppressed how much you got. So here you can see the strength of the connection, that's what the size of these dips mean in the control situation. Bigger as a result of anodal, smaller as a result of cathodal. But what you can notice here by eye, and is quantified here, is the anodal is more effective at producing boosting than the cathodal is effective in suppressing boosting. Once again, we see that because of endogenous processes to the brain, how the brain itself generates plasticity, it's easier for us to make more plasticity than it is for us to suppress plasticity. What does that mean at a global level? Once again, imagine we have a mixed polarization. Half the neurons are getting the inward, excitatory, half the neurons are getting the outward, inhibitory. What's the net effect of plasticity in that region? The net is a positive effect on plasticity. So again, it's not a zero-sum game. Now, the mechanisms of plasticity induction, how direct current affects it, is significantly more complicated than what I'm showing here. There are details in this paper. We've shown that the nature of the plasticity affects what these things do, but it is certainly the general case that the modulation of plasticity by direct current stimulation is not even for the anodal and the cathodal variants. Okay. The up to now, I've sort of talked about this mixed polarization as something that just happens. You just sort of have to deal with it. But we have ways to deal with it and to explain how a mixed polarization can still lead to a consistent effect. But we're not just passive observers to this current flow pattern. We can actually do things to control it. We can control how current flows through the brain. As I started at the beginning of this video on, we control it by where we put the electrodes. I want to go back to this experiment, and so this shows you all the potential places you can put electrodes when you're talking about HD or high definition electrodes. These are these smaller electrodes as opposed to the pads I showed you before. So in this particular study, in collaboration with Michael Nietzsche's group, we compared two different electrode positions. The first one is what I talked about before, the M1 SO montage, a large electrode over the motor region, another large electrode in the supraorbital position. We compared that against the 4x1 high definition montage, and I showed you the simulations of these before. Let me show you those simulations again. This montage here, the classic M1SO produces a relatively diffuse current flow. You can tell that the peaks are not necessarily under the electrodes, but between them, whereas the 4x1 produces a much more focal current flow. Now the peak intensities here are less than the peak intensities here, but in the region of interest, which is actually the motor strip, the intensities are actually the same. Another aspect or comparison between them is when you use conventional tDCS with large pads, the current flows deep. And this has to do with that notion of continuity from one region to the other, whereas with the 4x1 montage, we can be much more superficial. And now you can start to get a better sense that in the target itself, the intensities are comparable, and so we spare these regions. But when we look at directionality, right, we see the notion that in the case of the conventional montage, again, we're getting this zebra striped of inward and outward. We're getting a mixed polarity. And with the 4x1 montage, we can generate a more consistent polarity. And this is, if you remember all the way back when I was talking about the single electrode surrounded by the ring, it's based on the same ideas. Why is the 4x1 more unidirectional? To explain it simply, there is a center electrode and it's surrounded by other electrodes, usually 4, but in this case I'm just showing you 2. Now, if 1 milliamp is being delivered through here, because of the conservation of current, each one of these is only collecting negative 0.5. If I had 4 electrodes, if this was delivering 1, each one of these is only connecting negative 0.25. So you can see that the center electrode is sort of stronger than the other ones. And for that reason, as well as the fact that the current can actually come back through a longer pathway, we end up producing a more unidirectional effect with the 4x1 montage. And so if you're very concerned about directionality, the 4x1 allows you to control that directionality as well as where you put the stimulation. We can also think of directionality in a different way. This was a collaboration with the group of John Rothwell in the UK. Now this is a picture I showed you earlier. We have just two HD electrodes in this case. We have an anode here and a cathode here. And so the current is flowing in, and here we have inward radial, and here we have outward radial. It's mixed, but it's known. Meaning, if we have a particular region of interest here, in this case this is the motor strip, let's say we're interested in the hand knob of the motor strip, it's not that we don't know the directionality, we can actually control it. If it's on this side of the gyri, then putting an anode here can consistently give us an inward radial current flow. And so sometimes I call this sub-gyri targeting, because we can control the direction of current flow even into the sub-gyri level. Now, it doesn't mean we don't have mixed polarization everywhere else, but we can control it in the gyri level. And so I've just shown you very quickly 2 examples of how we don't have to just be sort of passive observers to this mixed polarization, but we can control it as well. So I hope in this talk I'll first introduce why tDCS produces a mixed polarization, right? I've explained to you how we can try to manage it, and I've explained to you how it's not a zero-sum game. Again, there are other videos I can point you to where I talk more about some of the foundations of tDCS and also deployments of things like high definition tDCS for more precise outcomes. So thank you for your attention.